So in our QTL, this plot has an argument upper that I can say is, or lower that I can say is, say, FV1, or, and then it will replace, instead of showing the LOD curve, the LOD scores for the 2QTL model versus the null, it'll show the, in the lower triangle, it'll give this comparison where it's comparing FV1 is the full model versus the best versus this best single QTL model. And what happens is it cleans up those tails quite a bit so that you can more clearly see that I only really care about 7 and 15 in this case. 15 with 14 is not really any better than just 7 with 14 is not any better than 7 on its own. I guess there are two reasons that I'm interested in doing this two-dimensional scan. One is I want to know about interactions. Are there loci that look to interact that I didn't see otherwise? Another is, kind of if I control for one major locus, do I see evidence for a second one? And then particularly sort of looking in each of these triangles along the diagonal of, are there chromosomes where there look to be evidence for 2QTL? Just the main trick that we use is this, for each rectangle in this big triangle of the two-dimensional scan, I take the maximum over that rectangle and then I subtract off the maximum sort of along the diagonal. So compare the best 2QTL model to the best single QTL model, either allowing for interactions or not. If I want to ignore interactions and just look for additive QTL model, I can use AV1 instead. So it'll just fit two QTL strictly additive model to a single QTL model. I could put, the two full versus the interacting versus one and the additive versus one on the other. I might, if I just focus on a pair of chromosomes, it'll be faster. But there's really good evidence for there being a QTL on both 7 and 15. If I put the interactive LOD score in the top, using upper equals int, we'll do the difference between the full and the additive model. There's really no good evidence for 2QTL, for an interaction between these loci when I just look at these LOD curves, LOD scores. The summary function is also useful for trying to figure out, trying to make sense of these two-dimensional QTL scans. It is hard to make sense of without having done the 2QTL permutation test, which is a hefty computational burden, but in this case I've already done for us. So if I do summary and then the two-dimensional QTL scan output, give it my permutation results that were very painfully obtained and a significance level. What it'll do is go through each pair of chromosomes, find the positions of the 2QTL under the full model where they're allowed to interact, the positions of the 2QTL under the additive model where they're assumed to be strictly additive, and then calculate a bunch of these LOD scores. The LOD score comparing that 2QTL interactive model to the null, the LOD score comparing the 2QTL interactive model to the best single QTL model, a LOD score for the interaction, which is comparing the best full model to the best additive model, and a LOD score comparing the 2QTL strictly additive to the best single QTL model. The permutation test it'll calculate each of these LOD scores at each of many permutations, and then here we'll get the 95 percentile of each of those specific LOD scores. And this function, the rule that we have for printing this out, is I look for these two things to be big or these two things to be big. Or these two things to be big. So I want either evidence for a second QTL, and this is evidence for a second QTL, or good evidence for an interaction. But with this particular data, the only row it gives me is this pair of QTL on 7 and 15. And the evidence for interaction is not strong. So remember, this is an intercross. So the test for interaction is comparing a 9-parameter model of the 3 genotype, the 9 possible 2-locus genotypes, all allowed to have different means, to a 5-parameter model, which is like the intercept and the 2 QTL effects, the 2 parameters for each of the 2 QTL. So the test for an interaction has 4 degrees of freedom, so it should naturally be kind of big. So a LOD score of 1 is not interesting. This does take an argument, p-values equal true. So it will use those permutations to calculate the proportion of permutation replicates that gave a full LOD score bigger than 12, or an FV1 LOD score bigger than 5.9. So you can see I have, really strong evidence for there being a second QTL if I assume they're additive. There's, no good evidence at all. All the permutation replicates, there was somewhere in the genome that had an interactive LOD score bigger than 1. So a p-value for the interaction here is 1. No good evidence for an interaction. I can't say they don't interact, but I don't have evidence for an interaction. And if I were to allow for an interaction, that's then a 6 degree of freedom test. And the p-value is not that interesting there. The interest is only if I assume that there's no interaction, because that's a fewer degree of freedom thing. Yeah?
So Trudy McCabe believes that all the interesting QTL interactions, epistasis, are in absence of main effects. You've sort of built the tool. So has that been what you've seen too, or is it usually QTL with main effects interacting?
Those are the interesting ones, or those are the most common?
Probably the most common is a better way to put it. Definitely.
I don't think that they're the most common.
Okay.
I think it's really unusual to have 2 loci that have an important interaction but have no main effect at all. So that would be, that the say in a back cross, that one QTL is say decreasing the phenotype and the other one is increasing the phenotype, and in just kind of, or at one, with one genotype, for one of the genotypes at this locus increases the phenotype, whereas for the alternate genotype at this locus, the opposite thing happens in a perfectly balanced way. I think is unusual. It's interesting. It's especially interesting, I think biologically. No matter what these things are, that would be really interesting. And it's interesting kind of from a methods perspective, because this is really, you have no ability to find these loci in a genome scan. If you're just thinking one QTL model, they totally cancel each other out. I mean, you can't see anything, or there's nothing. And so you'd have to look at two QTL models for it. But I think it's unusual. Most QTL will have involved in interaction will also show some marginal effect. In this example that I really like, there's a QTL on chromosome 1, on chromosome 4 that are really strong. The 6 and 15 loci that were marginally sort of next best but not very close, they have a really strong interaction effect. That this is a back cross, so the test for an interaction is just 1 degree of freedom, and the LOD score is like 4 for that interaction, which is big in this case and clear. You really only see those loci well if you account for that interaction. So for that data set, so this is the main summary that comes out, I guess, that if I look at 1 and 4, there's no real evidence for an interaction between those two loci, but really strong evidence for either 1 or 4. I mean, there's good evidence for there being 2, which is like not that surprising, I guess. For 6 and 15, if I allow for an interaction, the chromosome 6 locus is at 55 centimorgans. If I assume additive, the chromosome 6 locus is at 24 centimorgans. So the chromosome 6 locus shifts a lot. It's a different part of the chromosome than the main peak in the single QTL scan. And so LOD score for the interaction is, if I compare this model to the best additive model, the LOD score is 2.9, and it ends up being very strong evidence for it. If I look at chromosome 1, if I allow for an interaction, I mean, there's not really any good evidence for if there are 2 loci on chromosome 1, do they interact? It doesn't look like they interact. Whereas the additive model that maximizes at kind of the obvious places are those 2 big bumps that were in that double bumped curve. The evidence for a second QTL, given that there's a first, is not very strong. Yes?
What are your, what level of interaction? Like, when you say, so you look at a number, when you say large, so the interaction is strong, or the additive model.
So I do this permutation test, and then I look at the p-values associated with these.
Okay, so you wouldn't make an assumption just based on what you see right there, you would make the p-values as well?
I would, these LOD scores, what LOD scores are interesting, you have to keep in mind that these all have rather different degrees of freedom with them. And so what's big here versus here can be quite different. Really, the only good way to gauge is by having done that permutation test and looking at it, I think. Yeah, I didn't show them here, but this one is big, and if you do the permutation test, it's big enough to be, really trustworthy as a clear interaction. Yeah. So that I think there's not actually good evidence for a second QTL on chromosome 1 in this case. I think if it were if I were trying to decide, should I account for the possibility that there are two genes on that chromosome affecting the trait, I would say, there's a reasonably good chance that there are two. But if I wanted to say, but really the evidence for there being a second QTL is not very strong. Yeah.
So if there was evidence that there is a second chromosome, a second QTL on the chromosome that is affecting the trait, how would you determine the second QTL?
I use this approach. Do a two-dimensional scan over that chromosome and look at what is the best 2QTL model and how does it compare to the best single QTL model, which is this log score right here. And I would compare to how that behaves under in the absence of QTL. Here's for that chromosome 1 locus, this is for the additive versus the single QTL model down here. And it has the peak is at, a little above 1.5. And I compare that to what I think it would be if there were just one QTL. I think this is, interesting, but not very strong. Like I could say, a good chance that there are two here, but you could reasonably get this sort of data in the absence of, with just one QTL, I think. And if I look at the QTL effects, just looking at the pair 1 and 4 together, and there's a line right here and there's a line right here. This is sort of the effect of the chromosome 4 locus when you are homozygous at the chromosome 1 locus or when you're heterozygous at the chromosome 1 locus. Those two lines are pretty parallel. Maybe some of you can only see one of those two lines and just trust me, the other one looks right similar. So then I would say those two QTL do not look to interact at all. Whereas this is the 6x15. If you are homozygous at the chromosome 6 locus, there's basically chromosome 15 has no effect. Whereas if you're heterozygous at chromosome 6, chromosome 15 has a big effect. An effect that's really as big as any one locus involved here. The interaction going on at 6 and 15 are just as interesting in a way as the biggest locus from scan 1. In this case, if you don't consider the possibility of interaction, then you would miss these two really, probably the largest effect in the study. And the way you do these again, using scan 2 is just like using scan 1, but it takes longer, and interpreting it is confusing. Takes some study. The main things are really, the main thing I focus on is comparing the best 2QTL model to the best single QTL model for each pair of chromosomes at a time. Also look at the difference between the best 2QTL interactive model and the best 2QTL additive model. And I will most of the computations I do in a study will be two-dimensional scan permutation tests to get thresholds on these things.
Are you going to be talking about the penalties that you can calculate from the scan 2?
We've got 15 minutes before lunch, so I will try.
All of these models, they assume your phenotypic data follow a normal distribution, right?
Mhm.
So what happens if your data doesn't follow a normal distribution? What's your approach? You just transform the data and use another model?
My main approach is really, is to transform. The question is, if I have a distribution that's not normally distributed, what should I do? What we want is that the residual variation after accounting for QTL, that thing is normally distributed. So that most of the time I want a phenotype distribution that's sort of blob-shaped, vaguely normal. If it's really highly skewed, I will take logs. If it's, like count kind of data, I'll take square roots usually. And for gene expression data, I often will use normal quantiles to just force phenotypes to follow a normal distribution. Partly that if you're presented with 25,000 phenotypes, you're not going to go through them one at a time and figure out whether they're nicely behaved distributions. So I will just take the ranks, turn those into normal, the quantiles on the normal distribution, just force the marginal distribution to be normal. And that, I think, is not unlike doing a non-parametric kind of test. I think for these interactions, having done a transformation, the meaning of the interactions is totally screwed up. And outliers in the phenotypes can cause major headaches in looking at these two QTL models with interactions. But I'm mostly interested in finding the QTL and not so much whether they interact or not. And so I don't worry so much about the meaning of the interaction, but I do care about whether these are real QTL or not. But transformations, I basically choose between not transforming, taking square roots, taking logs, or forcing things to be just normally distributed.
And if I can add something, then please disagree. The important thing to remember is that it's your residuals that are supposed to be normally distributed. So if you have a major QTL effect, it's not going to be normally distributed when you put the data in, right?
Except for gene expression, most of the phenotypes I see don't have QTL effects that are so large that they would be obvious in the marginal distribution. Occasionally you see a bimodal distribution, but it's pretty unusual. And even with gene expression, you can have, close to Mendelian effects, but you won't see it immediately in the marginal distribution.
We discussed different ways of inputting your data into our field, and having it like format.
Mm-hmm.
All that. How would you recommend the best way to do that?
So I think maybe we should just create a list of things we want to cover and try to do it in the afternoon. So the main thing that I wanted to cover that I didn't quite get to, I didn't at all get to, is multiple QTL models beyond 2. There are various methods for exploring these multiple QTL models or functions for doing that. There's a stepwise QTL function that uses a particular set of penalties on size of the model. I want to cover that. But data formats, that, I mean, I'm happy to do that too. Yeah.
Going back when we did the multiple overlays and 4 types, is it just the coding only allows you to put 3 in at a time and you have to add 4 to fit on?
That's right.
Okay, so just only handle 3 at the initial plot.
That's right. For no good reason, really, now that you pointed out. Yes.
If you have many markers, you end up with fragments of like chromosome 1 being broken down to many different groups. Is there a way to say if where there's no QTL, it will not block that fragment and pick it up? Because if you have so many different groups, if you block them, you cannot see it on the top.
Yeah, I guess I don't have a good solution to that. But so that if one of your linkage group, I mean, if one of your chromosomes is in a bunch of different pieces and you don't know how they fit together and you do this kind of calculation, then the plots look terrible.
Yes.
Yeah.
In your case, you have I think 20 and X.
Mm-hmm.
So in my case, I would have like 70 of them.
Yeah, I don't have a good solution to that, really.
So for the two functions, scan 1 and scan 2, we do scan 2, you basically reanalyze all the data you do, like just like analyze those significant QTL you picked up from scan 1.
That's right. Scan 2, I mean, so Scan 2 does not look at just the loci that you saw with effect in Scan 1. It does a full exhaustive calculation. For the, I mean, for the, you know, the kind of old school data sets of 100 markers, or even if, you know, sort of typical marker sets when you have a plain cross of this type where you have, say, 2,000 markers, that is doable. If you have 20,000 markers, it does not work well. But that function will just do an exhaustive search. But I'll show you a little bit of how, I mean, you can explore what's going on, just, you know, sort of picking out the main QTL and then looking for interactions among them. I'll show that this afternoon.
What's the largest number of markers you can
I had an experiment recently that had 10,000 markers. I guess for some parts of the analysis, I had 80,000 markers. Some functions, it can handle 100,000 markers without any problem. Other functions, it can't handle 1,000 markers very well. So Scan 2, where I'm trying to exhaustively calculate all possible pairwise, you know, linkages, or I mean, 2 QTL models, that the size of the object you're trying to calculate is just too big, and that, like, the plots that you make are just you don't have enough pixels on a screen anyway. The genetic map estimation, I would say, you know, if you have 1,000 markers on a chromosome and an unusual pattern of missing data, or really kind of any pattern of missing data, re-estimating marker locations is deathly slow. But there are some tricks that can speed that up. Basically, anything where you're doing pairs of markers, then there's the limit of the number of markers you can handle is reached pretty fast. Anything where you're plotting like all possible markers of some kind, the plots look really terrible. But if you have already a clean genetic map and you want to do a genome scan with 100,000 markers, I don't see any reason why you couldn't do it. But the RQTL is really written for a back cross or an inner cross. The recombinant inbred line stuff is sort of added in on top. It basically just treats recombinant inbred lines like a back cross, but changing the distances. And for a back cross or an inner cross, more than 2,000 markers is way overkill. So, the design of the package doesn't really fit with modern situation.
So sometimes we use the other software like MapQTL to do the scan or analysis.
Mm-hmm.
Generally, if to pick 20 centimorgan away, we assume there are two QTL. So how do you think? Should we do some other analysis beyond that to confirm?
Is this in recombinant inbred lines?
Yes.
So in recombinant inbred lines, that's maybe a better bet because the kind of the map expansion makes it so that 20 centimorgans apart is more like 40 centimorgans apart or more. But I would prefer to fit multiple QTL models, to compare the two QTL model to a single QTL model. Okay, let's have lunch.